I'm not hearing anything.
Oh,
Is it just me?
yeah.
Well, I'm hearing
No.
you, but no, but not the talk.
Wait. No one can hear anything?
No.
I hear you, Michael.
Damn it. I hear everything.
But can't you run
Sorry,
it through your computer? I mean, if you take your ears off.
I'm running it.
I think you have to activate the audio.
Well, that's what I hoped I did.
Key option, you have to do so.
Because of the timing of my my new baby's arrival. And so thanks for bearing with me. I hope to be there live for the Q&A session. So I'm going to talk to you today about immunogenetic adaptation to disease in one particular amphibian species, but I wanted to start more broadly by talking about the problem of emerging infectious diseases across different ectothermic vertebrates. And so I've thrown a handful of examples up here. Of course, there are many more. I've got chytridiomycosis in frogs, which is what I'll be talking about. We've got ranavirus, which infects turtles, fish, and amphibians. Snake fungal disease, which is caused by a fungal pathogen, sea turtle fibropapillomatosis, which is linked to a herpes virus, upper respiratory tract disease in tortoises. And for those of you who are confused by the fact that I have a bat up here, which is an endothermic mammal, when bats get white nose syndrome, which is a fungal disease, it's when they're hibernating in the winter and functioning more like ectotherms. And so I think it's intriguing that these organisms that require external temperatures to maintain physiological processes such as immune function are having particular challenges with emerging infectious diseases. And what I think this really calls for is that we really need to fill some fundamental immunology knowledge gaps, because compared to endothermic birds and mammals, we know very, very little about how ectothermic vertebrate immune systems function, evolve, and how they're even genomically organized. So with that plug, I also want to introduce myself as what I call a lifelong frogologist. That's what my family likes to call me. So I've always had a personal passion for amphibians. I've always really enjoyed frogs as organisms. I now like to share them with my children. And because of this passion, and because there have been disease threats in amphibians pretty much my entire lifetime, that's driven a large portion of the focus of my research career. So specifically, I study chytridiomycosis, which is a pretty nasty disease in amphibians. So I know dead frog photos aren't the nicest to look at, but I just highlight them here so that you can get a sense of really how dramatic and devastating this disease can look like in the field. Amphibian chytridiomycosis is a disease that we have extensive experimental and field data on at this point, decades of research, so I could tell you all kinds of things about it. For the sake of time, I wanted to hone in on a few key features. It's a global disease that has recently emerged. It is specific to amphibians, so it doesn't infect any other vertebrate group. It's caused by the fungus Batrachochytrium dendrobatitis. We now know that there is a second species, B. sal, that is salamander specific, but I'll be talking about BD in frogs in my talk today. And the final thing that I think is really fascinating about BD is that across different host frog species, sometimes it causes extinction. In many cases, we see species going extinct, populations being extirpated, and major die-offs and declines. But then there are as many, if not more, other species that readily get infected with BD, and it appears to be largely benign. It may be causing some sublethal consequences. And so what kind of drives that difference in susceptibility from a host perspective is one of the major research questions that I've focused on throughout my career. More specifically, what I've been doing is honing in on the frog genus Rana, particularly the North American clade, and looking at how among these closely related species, we see the complete range of 100% susceptibility all the way to 100% disease resistance or tolerance. So we have 5 species that I've chosen here. There are a few dozen others I could have put up there, but I happen to have active research projects going on with these 5, and we know roughly where they fall on this disease scale. I think it's really fascinating that within a pretty close evolutionary history, we have some species that all individuals are susceptible if they get infected with BD, ranging to the North American bullfrog, which shows complete BD tolerance and really seems to suffer very little negative consequences of infection. But then we do have species that fall in the middle. And so these variable susceptibility species are where we can ask some really interesting questions about what causes differences in susceptibility. And so what I'm going to tell you about today is specifically research that we've been doing within one of these species, Rana javapaensis, which has been my focal study species for the past 15+ years. And so we've generated quite a bit of data characterizing that it is quite susceptible, but not entirely so. And we see some populations and individuals that show evidence of BD tolerance. So a little bit more about Rana javapaensis. This is the lowland leopard frog. It is endemic to Mexico and Arizona within the United States. The Mexico populations are very poorly characterized, and so I'm focusing on the US distribution in my work. We can say that BD die-offs have been happening for about 30 years because that is when they were first documented by state biologists that were doing routine surveys. And so we kind of have a distinct time point in the late 80s, early 90s, when there was a major change and we started seeing these die-offs happen for the first time. Now, one of the major ways that I've started looking at host susceptibility in this species is by looking at major histocompatibility or MHC class 2 genes. The schematic diagram on the right is kind of a cartoon showing you how important these MHC gene products are for acquired immune responses. So antigen presenting cells express MHC class 2 proteins. What they do is they present pathogen peptides to T cells. And this interaction is essential for stimulating any kind of acquired immune response with memory and very specific response to a particular pathogen. And so because of this key interaction, MHC class 2 genes are really good candidates for looking at any levels of sort of association with genetics and disease susceptibility. And in fact, there are hundreds of other studies in vertebrates throughout the literature showing many relationships between MHC genetic variation and disease susceptibility. So that's exactly the approach that I took for the first decade of working in this study system. And unfortunately, I don't have time to go into any of the detail here, but this is just highlighting that I've done a number of previous studies with collaborators where we've established that based on field mortality data and field genetic data, as well as experimental infections in the lab, MHC genotypes drive which individuals can survive BD infections and which individuals can't. So that's sort of the background context to start telling you about some of the newer work that I've been doing. And the latest study we've published has focused on gene expression profiling of the immune response to BD infection. So I'm showing you a picture of one of the egg masses that we collected for this study. We collect egg masses from different genetic populations in the field. Eggs cannot be infected with BD, so we know they're starting out as naive. We then raise them up in the lab all the way through metamorphosis, and then we take our frogs and we expose them to pathogenic BD in the lab and track whether or not they survive. So the particular study that we did here used RNA-seq across different immune tissues to look at what were the immune gene expression patterns that significantly changed in frogs that were susceptible to BD or were not. And we looked at skin, which is the site of BD infection, and spleen, which is the major immune generating tissue in amphibians. For the sake of time, I'm only showing you the spleen results, but the skin results are similar, if not quite as strong. And to orient you to what I'm showing you here, I'm only showing you immune genes that showed significant differences in expression between different treatment groups. And then I've broken these genes roughly into those that are part of sort of innate immune function, inflammatory responses, and then acquired immunity. So note that we've got class 1 and class 2 MHC in this category. And then the way that we grouped our individuals is that we had 33 control animals that were sham infected. We had 20 that we sampled at early infection stages before disease signs manifest, but we kind of wanted to see the early response. And then the frogs that survived into the late stages of infection, which is when chytridiomycosis manifests, if it's going to. We had 19 that survived the whole experiment without any disease signs, and 9 that were susceptible and developed chytridiomycosis. And so those are what we compared. The red colors indicate significant gene upregulation or increased expression, whereas the blue indicates significant decreased gene expression. And so the major pattern we found here is that if you look at those susceptible frogs, we see a whole lot of red, a whole lot of increased gene expression, particularly of the acquired and inflammatory genes, including MHC. If you compare that to the surviving frogs, they actually show a very strong blue response, which is decreased expression even relative to background levels and lower than the control uninfected individuals. So to make this perfectly clear, this was the exact opposite pattern of what I had expected to recover. The thought going in was that frogs that were surviving were probably ramping up acquired immune responses and mounting more potent responses. But instead, we see the exact opposite pattern, that survival is associated with downregulating immune gene expression. So this was a surprising result, but there are ways in which it actually makes perfect sense. And so our working hypothesis is to say the more frogs are fighting BD, maybe the worse it gets. So maybe this interaction where you initiate an acquired immune response and ramp up that branch of the immune system, we know this interaction matters, but maybe not in the way we thought it did. Maybe it's actually the frogs that don't present BD to T cells, that don't have MHC alleles that are particularly reactive, that actually manage to survive their infections better. And one way in which this possibly makes sense is based on what we know about BD as a pathogen, which is that it's immunosuppressive. So we have good data now that BD secretes a compound that causes amphibian T and B cells to undergo apoptosis. So maybe generating these immune cells that BD can just readily kill is just this energetic wasteland that ends up being counterproductive. So that's where we're at in terms of the functional immune response. What I want to spend the rest of my time telling you about is broadly looking at whether and how the rest of the Rana javapensis genome is adapted to BD and chytridiomycosis. And so this is work led by Dr. Kevin Mulder, showed in the picture here. So I was Kevin's PhD co-advisor, and he successfully defended his thesis just about eight or 10 months ago. So he's the newly minted Dr. Mulder. And one of his doctoral thesis chapters focused specifically on this question about what can population genomics of the functional part of the Rana javapensis genome tell us about adaptation to BD. So if we look at this map on the left, this is showing you the North American distribution of Rana javapensis. Every symbol is one of the populations that we have studied in detail and that we did genomic characterization for this study. And before I get into the results, what I want to do is hone in on some key features of these populations that are important for understanding how we did our analyses. So we know that populations differ in their BD tolerance or susceptibility levels. This is based on that prior field and lab work that I've done that I had briefly mentioned. And so the different symbols here show you different disease susceptibility categories. So for example, the X's are completely susceptible populations that show 100% mortality in the field and lab, whereas squares that are tolerant show complete ability to survive BD infection, and triangles are variable, meaning it's at the individual level. Some individuals survive and others don't, and these populations are likely evolving tolerance to BD. The remainder of the population shown in circles are, we don't have enough data to say definitively about susceptibility, so we just kind of leave them as unknown. The next thing I want to mention is that 2 of these study populations have gone extinct since about the year 2010. So, in my 15 years of annual surveys of these populations, 2009 was the last time we saw frogs at either site, and we have good evidence that they are now extirpated. So they've done poorly enough that they are actually no longer there at all. And then the final thing I want to mention is this one site shown in green, which is a very unique population. It's called mule shoe. And frogs at this site live in a natural hot spring where the water is so warm that it shields the frogs from BD. There is BD in the area, but the frogs never get sick, and it's a very robust population because of this warm water, either keeping BD from being able to grow or enhancing the frog immune function, or maybe some combination of both. So to tell you just a little bit about our methods, this was a sequence capture approach. So we developed a sequence array based on a combination of neutral genes as well as candidate genes. And so what I mean by that is we took our own RNA-seq data as well as information from other studies in the literature to identify over 800 candidate genes that show differential expression in response to BD infection. In addition, from our RNA-seq data set, we took just a random set of 579 genes with SNPs. And so we call that presumptively our neutral data set. And then we also included 2 mitochondrial genes for comparative purposes. In terms of the lab work, we sequenced 134 individuals total across those populations that I just told you about. This was all Nextera style library prep and high-depth NovaSeq sequencing. I'm not going to go into the specifics of our bioinformatics or other programs we use, but for those who are interested in pipelines, I wanted to put it up here that you can see as a reference. And as I go through our results, I will touch on each method that we used as we use it. So to start out with just overall population structure, I'm showing you the map of our populations again on the left, where the colors match in terms of the geographic location and then the genetic variation. And the reason I'm showing you the map is because I want you to see how closely our PCA analysis of all of our genetic variation matches the geographic distribution of populations. So we've got this group of northwestern populations shown in the cooler colors. This gap in the middle, by the way, is because of Phoenix and all of the urban development. And here in the PCA, we see that all of those populations are clustering pretty tightly together, with the spread on the x-axis roughly matching the location of the populations. And then likewise, these other southeastern populations are all clustering over here on the right. And again, we see roughly the south-north spread of those populations matching the y-axis here. So pretty strong evidence of a phylogeographic component to the distribution of genetic variation. Next, I want to tell you a little bit about the phylogenetic results. And so this was a RAxML phylogeny based off of 88,000 base pairs of data. Using a closely related species as the outgroup, and we recover all very well supported clades. Every population is genetically distinct and falls into its own clade. We also see these warm color or cool color northwestern populations falling into the first major break, and then the second major clade is all of those southeastern populations, so consistent with that PCA analysis. And then finally, we group these populations into whether or not they are BD tolerant, variable, and susceptible. And we did this because we really wanted to ask the question, you know, has tolerance evolved multiple times in different populations, or could we say maybe it has a single origin within a particular clade? And the answer really thus far is that it has to have evolved multiple times. First of all, BD has only been impacting these populations very recently, since they have been isolated, distinct populations. But even beyond that, we see different susceptible populations falling into the upper and lower clade, as well as different tolerant and intermediate susceptible populations. So we do think there's the potential, at least across multiple of these populations, for tolerance to evolve. So next, I want to show you our admixture results. This is based off of over 2,000 SNPs. And what we see here actually really nearly identically recreates what we found with previous earlier microsatellite work, only we just have a lot more data going into it now, which is that, again, every population is genetically quite distinct, falling into its own genetic deme with nearly 100% assignment of the genome of all individuals. So overall, we have very strong nuclear-based population structure. The interesting thing is that when we look at the mitochondrial data, we see a completely different result. There's very limited mitochondrial genetic diversity. We don't have that many haplotypes across all of these individuals. And we don't see any structuring whatsoever based on the major geographic regions or based on the populations. And so this discordance is intriguing, and we have a little bit of insight into what's going on here. One thing that's been proposed previously from earlier mitochondrial work is that likely the US distribution of this species represents a northern expansion from Mexico, so maybe that's why there's limited mitochondrial diversity. We know that there has been water flow change, restriction in stream flow, and disease that all have likely contributed to fragmentation and isolation of populations, as well as strong population declines. And so essentially, we think all of these things are interacting to produce this unique pattern of currently every population is very genetically isolated because of strong genetic drift in small populations and extreme isolation. But overall, we see that this has been a pretty recent expansion throughout this entire region. And what we're really doing now are some better demographic analyses looking at historical population sizes to try to get a more precise answer, especially across these different populations. So next, I want to talk more specifically about how BD has impacted genetic diversity in these populations. So I'm showing you two metrics of genetic diversity, heterozygosity and allelic richness. And the populations are ordered the same way in both panels, which is from lowest disease susceptibility down to highest disease susceptibility. So in yellow, that control screen population where BD is not impacting them at all. Then we have the two tolerant populations, the two variable susceptibility populations, the three completely susceptible populations, and then finally, one of the unknown populations, the only of the unknown populations where we had large enough sample sizes to really do this analysis well. The next thing I just want to mention is that the two shown with black outlines are the two populations that have been extirpated, where one is unknown susceptibility, but one of them we know is a very BD susceptible population. And so again, I've ordered these populations from lowest to highest BD susceptibility, but what we see is that the metric of genetic diversity almost exactly matches that pattern, where the less susceptible populations have higher heterozygosity and higher allelic richness, and the more susceptible populations all have the lowest metric of genetic diversity. The third thing I want to tell you about are the number of private alleles across these populations. And I want to emphasize that it's a really different pattern. So here we're not seeing that the less susceptible populations have more private alleles. In fact, some of these highly susceptible populations are harboring a lot of private alleles. And sadly, the two that have been extirpated, one of them in particular had the highest number of private alleles recovered. So highlighting this was a really genetically distinct population that we have not lost. So this is sort of a conservation argument that as we're losing populations, we are really losing a lot of important functional genetic variation in this species. So just to emphasize the major pattern here, both heterozygosity and allelic richness decrease with BD susceptibility, and there's a strong loss of genetic diversity in this species overall. So now on to looking at specific patterns of genes under selection. So what we did here is we took three different metrics of outlier analyses that provide evidence of selection. We looked at FST outliers from our Bayes scan analysis. We looked at PN-PS ratios, which is equivalent to DNDS. So basically, are there elevated non-synonymous to synonymous substitution rates at the molecular level? And then we also looked at the distribution of Tajima's D values. So just to give you a little bit more info about sort of what that looks like, if we take all of our Tajima's D values across every gene we looked at, we only would retain those that were at least one standard deviation below or above the mean as possible extreme values that indicate selective events happening in those populations. And so unsurprisingly, when we do this analysis, we don't see a ton of overlap between the three metrics. You know, with any type of outlier analyses with a lot of loci, you're going to get some spurious associations. And so it's exactly why we only focused in on the overlap between all three of these approaches. And when we do that, what we see is that there are 6 genes that are significant outliers across all 3 metrics. The 4 that are not shown in bold are not particularly important for immune function, and so we haven't investigated them in too much detail. But 2 of them are quite intriguing in that one is a T cell glycoprotein gene, and then another one is an immunoglobulin gene. So we have evidence of T cell and B cell genes being under selection across a variety of different metrics. And we now have 2 new candidate genes for some follow-up studies. Finally, I want to talk specifically about the GWAS outlier analyses that we did. So first, we used Bayescan to look at FST outliers. And what we did here is we grouped populations by those BD susceptibility categories and then looked for associations between particular SNPs and susceptibility. I really want to emphasize that the really strong nuclear population structure we have very much reduces your ability to detect outliers with these particular methods. But we did find a couple, and one in particular I want to talk about, which is IL-10 or interleukin 10. This is a major inflammatory gene that's involved in innate and acquired immune responses. And interestingly, when we look at this relationship, we have 2 SNPs, A and B. And when we look at the frequency of these SNPs across BD infection categories, we see that individuals with medium BD intensity predominantly have allele B and not A. And because medium BD is kind of the level that could be associated with BD tolerance, where you're infected, but you're managing your infection, we think this might be a candidate resistance allele. Finally, we used zambada as an alternative GWAS method. And the reason we did this is because zambada allows you to use continuous data, continuous variables. And so here we simply grouped individuals based on their actual BD infection intensity values. And then we're able to look at significant outlier SNPs that were associated with different levels of BD. And again, our strong population structure limited our outliers, but we do see 3 regions of the genome showing a relationship. And in particular, we're interested in this locus right here, based on what we see in the BD data. So at this Raya 425 locus, we have individuals that are either heterozygotes or homozygotes, and the heterozygotes have significantly lower infection intensities compared to the homozygotes. So we think that that new SNP that exists in some individuals is again a potential resistance allele. Now, this gene is a Rab35 inhibitor. It has a role in exosome secretion. We don't see a lot in the literature about immune function, and we don't know that much about it. And so it's something that, once again, we need to follow up on with some functional analyses. But we now have this nice set of 4 genes that we can try to start doing some of that work in. So to try to quickly wrap things up into some conclusions that you can hopefully make sense of, we first looked at the gene expression data and found that the MHC and expression patterns were surprising, but we think based on these results that BD tolerance is maybe actually driven by MHC alleles that are not presenting BD to T cells or are maybe very immune regulatory. When we look at our population genomics, we see discordance between the mitochondrial and nuclear markers that gets into the demographic and evolutionary history of the species. When we look at genetic diversity, we see that either BD is impacting the diversity or the genetic diversity is driving BD susceptibility patterns. But we certainly see a relationship between disease susceptibility and how much genetic variation we recover. We see many signals of selection on many coding sequences across the methods that we use, but the overlap is limited to only a few immune genes that are now our latest set of candidate genes. And then finally, the FST outlier and other GWAS analyses are complicated by really strong population structure, but we find a few additional candidate immune genes there for now conducting some follow-up functional studies. And so with that, I would like to just briefly thank all of the funding sources, institutions, and people from the different lab groups involved in these projects who supported this research. And please email me if you have any questions, or hopefully I'll be there in person to answer your questions there. Thanks very much.